Hey you all, welcome and welcome back to the part 2. We have few topics left to be covered in this chapter, which are as follow: nucleic acid, primary and secondary metabolites, nature of bond linking monomers in a polymer, dynamic state of body constituents, which is the concept of metabolism, and metabolic basis of living, the living state, and enzymes. So let's begin each one of them one by one. Let's begin with nitrogen bases. Now see, living organisms have a number or a huge number of carbon compound content in it, in which many of them also have heterocyclic ring in it. Out of all these carbon compounds, some of these are nitrogen bases also. What are nitrogen bases? So these are the organic heterocyclic compounds that contains nitrogen and act as the fundamental unit of genetic code. Means they determine the genetic code by forming the sequence in DNA and RNA. Now, nitrogen bases are of two types. The first one is purine with the double ring structure, and the second one is pyrimidine with the single ring structure. Let's talk about purine first. As you can see on the screen, we have two examples like adenine with the abbreviation A and guanine with the abbreviation G. See, you can't just waste your time on lectures. You have to draw these structures by your own. Try to draw it, and if you get wrong, then try to balance with the hydrogen atoms, then you will get it right by your own. So the scientific name for adenine is 6-aminopurine. See, when you start numbering from 1 to 6 and so on the sixth carbon, you have to replace hydrogen atom with NH2 amino group. Then you will get adenine. All these numbers are marked in blue color, as you can see. And similarly, the scientific name for guanine is 2-amino-6-oxopurine. See, oxo is automatically, it means ketone group. Now, if you start numbering from 1 to 6 and so on, then on the sixth group you have to replace it with ketone group, and on the second amino means on the second position you have to replace it with NH2 amino group. Now we have pyrimidine with the single ring structures. Examples like cytosine with the abbreviation C, thymine with the abbreviation T, and uracil with the abbreviation U. Now the scientific name for uracil is 2,4-dioxypyrimidine. Means dioxy means di means 2 and oxy means ketone group at 2 or 4 carbon positions. So what you have to do is on the second and fourth position of carbon you have to balance it with ketone group, then you will get uracil. And the another is thymine with the scientific name 5-methyl uracil. You know why we have uracil in its name? Because its structure is as similar as uracil, but the only difference is because of the functional group methyl on its fifth carbon position, and the other structure will remain same as uracil. Now the third one is cytosine with the scientific name 4-aminopyridine. So what you have to do here is on the fourth carbon you have to balance it with NH2. Taking it in a very easy way with the respect of uracil, what you only have to do is try to balance fourth carbon position with amino group instead of ketone group, and you are all done. And you need to remember the structures of adenine and uracil. You can't ignore it because it is in your NCERT also. Our very first topic is nucleic acid. If you see in any living tissue, nucleic acids comes in under acid insoluble pool because it's a biomacromolecule. Similarly, as you read in the last session, polysaccharides, which are carbohydrate, and polypeptides of proteins, these all and these nucleic acids, these comprise the true macromolecule fraction of any living tissue or cell. Nucleic acids is a large biomolecule made up of long chain of nucleotides and its monomer like DNA and RNA that store and transmit the genetic information to the living organisms. And as the name suggests, nucleic acid, that means it's acidic in nature with the negative charge on it. Okay, so you may be familiar of this topic nucleic acids from your junior session from ninth class, I guess. So what are nucleic acids? See, passing of inherited traits from one generation to the another generation in the form of DNA, which is present inside the nucleus of cell. These DNA called the nucleic acids, and we already know it very well. So on a broad classification you will see that nucleic acids are of two types. One is DNA, which is deoxyribonucleic acid, and another is RNA, which is ribonucleic acids. So the nucleic acids can be found in, it is very obvious, you can say it by your own: nucleus, cytoplasm, mitochondria, and ribosomes. Why the cytoplasm, mitochondria and ribosomes? Because they are the semi-autonomous organelles of the cell. Now, nucleic acids are also heteropolymers, which means they are the polymer of different type of nucleotides. Now the question is, what are nucleotides? See, in human body, protein is the building block element in our body. Similarly, nucleotides are the building block element of nucleic acids. And also they are the monomer of DNA and RNA. See, in amino acids are the monomer of protein, and we already know this. Similarly, nucleotides are the monomer of nucleic acids like DNA and RNA. And these nucleotides are comprised of sugar, phosphate group and nitrogen base. Now we'll see what are these in the upcoming slides. Okay, so this is just an overview of what we are going to study now. So first we have nucleotides. Nucleotides has three chemically distinct compounds. One is a heterocyclic compound which is nitrogen base. We already know that. Second is a monosaccharide which we will cover in the upcoming slide. And third is a phosphoric acid. When all these nucleotides came together, they will form nucleotide chain, which is now a polymeric substance. And together they will form polynucleotide chain also by the help of phosphodiester bond between them. Two polypeptide chains are sufficient for the formation of DNA. We also have a factor of polarity on nucleic acids. For the example, we have DNA. In double standard DNA, the two strands are anti-parallel to each other. One runs 5 to 3 and the other is 3 to 5. You can see on the screen its polarity varies from 5 to 3 and other ends 3 to 5. Polarity is directly means to the direction, you know. First component of nucleotides is sugar, obviously, which is a monosaccharide because we have ribose or deoxyribose, which are the simplest sugars. They are 5 carbon aldose ring because they have aldose group in them. They are the heterocyclic compounds and also furan ring structures because of the 4 carbon inside the ring and 1 oxygen atom, you can see. So the first one is beta D ribose sugar with the general formula C5H10 and O5, which is generally present in the RNA, and the other is beta D 2 deoxyribose with the general formula C5H10O4, which is in the DNA. And why we have beta D 2 deoxyribose? Because there is only one difference between ribose and deoxyribose is because of the oxygen atom. You can clearly see I have mentioned in the red color, and even you can see in the formula deoxyribose has only 4 oxygen atoms. That also became the reason why ribose has higher molecular weight as compared to deoxyribose. So overall what you have to remember here is ribose helps in the formation of RNA and deoxyribose helps in the formation of DNA. You can remember it by ribose starts with R and RNA starts with R, and similarly deoxyribose starts with D and DNA starts with T. Then the second one is phosphoric acid or phosphate group with the general formula H3PO4, and they will be present in both the structures of DNA and RNA. Now the third one is nitrogen base, which we have already covered in detail, but just for reminding you, adenine and guanine are substituted purines while the rest are substituted pyrimidines like cytosine, uracil and thymine. We have already done their structures in detail. That will be sufficient for your chemistry also. So, and the other thing is DNA will have only 4 nitrogen bases like adenine, guanine, cytosine and thymine, and on the other hand RNA will have adenine, guanine, cytosine and uracil. Thymine and uracil are specific in both of them. Now we have nucleosides. So when you add sugar to the nitrogen base, you will get nucleosides. So what you have to do here is, first you need to draw the structure of ribose sugar. Then we need to add adenine nitrogen base to its structure. Let's closely examine this structure. On the C1 carbon position of sugar, we have OH group that will form the glycosidic linkage with the another NH group with the N9 position of adenine. Lose a molecule of water by this N-glycosidic linkage. By this we will get our first nucleoside, which is adenosine. Don't be confused with the glycosidic linkage. Here we have N-glycosidic linkage where N stands for nitrogen, which is between one carbohydrate and another nitrogen base. It's not O-glycosidic linkage. It is completely different, which we have dealt in the last session. It was O-glycosidic linkage where O stands for oxygen atom. which was between two carbohydrates. Examples like in the disaccharide or polysaccharide, any of these like malt sugar, milk sugar, saccharose, and any of these starch, but they both are different with the different perspectives. And by following these same steps with guanine, cytosine, and uracil with the same ribose sugar, we will get our rest of the nucleoside structures like guanosine, cytidine, and uridine. But you only need to remember the structures of adenosine and uridine because it's in your NCERT also. The only thing which you need to remember while drawing the structures of pyrimidine is the number of carbon and nitrogen is different as compared to purine. They vary accordingly. Ninth number of nitrogen links with the first carbon of sugar in purine, and first number of nitrogen link with the first carbon of sugar in pyrimidine. We can also do the same with the deoxyribose sugar. The steps will be as similar as the ribose sugar. So what you have to do here is by adding adenine with deoxyribose sugar, we will get deoxyadenosine. Similarly, we'll get deoxyguanosine, deoxycytidine, and deoxythymidine with both of the purine and pyrimidine nitrogen bases. And yes, these nucleosides with deoxyribose sugar is definitely not in your NCERT. I've just covered it additionally. So on the basis of your understanding, I'm going to ask you two easy questions. So the first one is there any deoxyuridine found in DNA? And the second one is there any ribose deoxythymidine found in RNA? You can take your time, pause the screen, and can answer me. So the answer is no. There is no deoxyuridine in the structure of DNA, and there is no ribose deoxythymidine in the structure of RNA, because in normal DNA the 4 strands which we already know are ATGC, which means adenine, thymine, guanine, and cytosine, and in normal RNA the 4 strands are AUGC, which means adenine, uracil, guanine, and cytosine. Now we have nucleotides. So don't be confused with the nucleoside and nucleotide. Side and tide both are different things. Now let's begin with the nucleotides. So when you add sugar plus phosphate group, you will get nucleotides. So what you have to do here is, I'm not going to explain the previous topic like you have to add sugar to the nitrogen base to the glycosidic linkage, those bonds, and blah blah blah. I'm not going to explain any of these things. Firstly, you have to add sugar to the nitrogen base by your own self. Then what we'll do here is, now we are not going to add sugar to the nitrogen base, then to the phosphate group. We are not going to do that because it will take a lot of time of ours, and we are smarter kids, so we'll do it smartly. So firstly, we already know that when we add ribose sugar to the adenine nitrogen base, we'll get adenosine. So we'll draw the structure of sugar plus nitrogen base, which is adenosine combinedly, and then we'll form the bond with phosphate group. Now look carefully, what I'm trying to say here is, this OH is the acidic group of this phosphoric acid, and this CH2OH is the alcoholic group of this sugar. So when this OH bonds with the alcoholic group of the sugar, one molecule of water will release out. By the ester bond, which we already know, when an acid and alcohol bonds together, ester bond forms. But this is not a basic ester bond, but it is actually phosphoester bond. I will explain it why. See, when the one molecule of hydrogen will release out from this phosphoric acid in the form of water, it will become phosphate. So we'll call it phosphoester bond. And then by following all these steps, we will get our first nucleotide, which is adenylate acid. It's called adenylate acid because of the phosphate group present in it. And again, by following the same steps with guanosine, cytidine, and uridine with the same ribose sugar, and of course the common phosphate group, we will get our rest of the nucleotide structures like guanylic acid, cytidilic acid, uridilic acids. And you only need to remember the structure of adenylate acid because it's your NCERT, and to make sure, I have already covered this before. We can also do the same with the deoxyribose sugar. The steps will be as similar as the ribose sugar, and we will get our deoxyadenalic acid, deoxyguanilic acid, deoxycytidinalic acid, and deoxythymidanilic acid. And none of these deoxyribonucleotides in your NCRT. Even I don't know why I'm explaining all this, for the sake of my own happiness maybe. But if these things are going out of your head, you can completely ignore it. Okay, so now we will classify nucleotides on the basis of number of phosphate group in it. See, basically there is no as such rule that you can add only one phosphate group to it. See, we can add 1 to 2, 3 phosphate group to the nucleotide structure. So we will see how we're gonna add and the structure, the whole procedure. We will simplify it in a very simple way. Don't be confuse anyone. I'm gonna simplify it from the basics. Now look carefully, what I'm trying to say here is, this is our pentose sugar with the R functional group on the second carbon position to it, where you can put OH group for ribose and H for the deoxyribose, which we already know. And on top of that, this is our base in the orange color. Here I have like only written base here. You can put any of the single or double ring nitrogen base structure to it, but for me, let's take, suppose we're taking A, which is adenosine. Now we will add our first phosphate group to the sugar structure. See how. Here we have CH2OH, which is an alcohol, and in the phosphate group we have OH, which is acidic in nature. The bond will form between these acidic and alcoholic functional groups by the help of phosphoester bond, and leads to the formation of our first nucleotide, which is AMP, adenosine monophosphate. And then we will add our second phosphate group to the previous phosphate group by the help of phosphoanhydride bond. You know why? Because in this phosphate group we have OH, which is acidic in nature, and the first phosphate group we have OH, which is acidic in nature, which leads to the formation of our second nucleotide, which is ADP, adenosine diphosphate, by which ADP forms, which is adenosine diphosphate. And similarly, the third phosphate group bonds with the second phosphate group by the help of phosphoanhydride bond, which leads to the formation of ATP, which is adenosine triphosphate. Okay, so let's end up all together in a conclusion. See, a nucleotide contains at least one phosphate group, which we all know. Now, depending on the number of phosphate group attached to the nucleotides, they are typically of 1 to 3 types. They are classified into 3 different types. See, the first is nucleotide monophosphate with one phosphate group. Example is AMP, GMP. The second one is nucleotide diphosphate with a 2 phosphate group in it, with the example ADP, GDP. Then we have nucleotide triphosphate with the 3 phosphate group in it, with the example ATP and GTP. And I guess you may know this phosphoanhydride bond before. You may hear it, because when two acids combine together, this bond forms by the loss of one water molecule in between them. And these two phosphoanhydride forms the higher energy bond as compared to phosphoester. So here we have a classification of sugar, nitrogen base, nucleotides, and number of phosphate groups. So here first we have both the sugars, which is ribose and deoxyribose. Then we have all 6 nitrogen bases. So when sugar plus nitrogen base combined together, example ribose plus adenine will become adenosine, ribose plus guanine will become guanosine, ribose plus cytosine will become cytidine, ribose plus uracil will become uridine. And similarly, deoxyribose plus adenine will become deoxyadenosine, deoxyribose plus guanine will become deoxyguanosine, deoxyribose plus cytosine will become deoxycytidine, and deoxyribose plus thymine will become deoxythymidine. And for nucleotides, when adenosine plus phosphate group combine, it will become nucleotides, like when adenosine and phosphate group will become adenylic acid, and guanosine plus phosphate group become guanylic acid, cytidine plus phosphate group will be cytidalic acid, uridine plus phosphate group will be uridalic acid. Similarly, deoxyadenosine will be fifth phosphate group will be deoxyadenylic acid. Deoxyguanosine with phosphate group will be deoxyguanilic acid. Deoxycytidine plus phosphate group will be deoxycytidalic acid, and deoxythymidine uh, plus phosphate group will be deoxythymidalic acid, where uracil and thymine are specific in both of them. So what I mean by the number of phosphate group is, when adenylic acid has one phosphate group, it will be AMP, which is adenosine monophosphate. When it has two phosphate group, it will be ADP, which is adenosine diphosphate. And when there will be three phosphate group, it will be adenosine triphosphate. Similarly, this list goes up to guanylic acid, GMP, GDP, GTP, and for cytidalic acid it will be CMP, CDP, CTP. For uralic acid it will be UMP, UDP, UTP, and for deoxyadenylic acid it will be DAMP, DADP, DATP, and DGMP, DGDP, DGDP, DCMP, DCDP, DCTP, DUMP, DUDP, DUTB. Okay, so by following this way, you can draw any of the following structures. Like suppose if I ask you to draw the structure of CTP, so what will you do here? First, you need to draw the three number of phosphate group to the structure of cytidalic acid, and here you're done with your CTP very easily. And what if I ask you the number of phosphate group in a nucleoside? What will be your answer? So the answer is 0. The number of phosphate group in a nucleotide is 0, obviously, because you can add phosphate group to the structure of nucleotide only, but not to the nucleoside. So it's no. Now we will see the formation of dinucleotides. So as you can see, this is our first nucleotide, and this OH group, which indicates that it is ribose sugar, and A on the nitrogen base, which means it is adenine. So this compound will be AMP, adenosine monophosphate, and this is our second nucleotide with the group O on it, which indicates that it is deoxyribose sugar with T on nitrogen base, which means it's thymine, and it will be DTMP compound, which means deoxythymine triphosphate. So when you will add these two nucleotide compounds together, it will become dinucleotide. So let's see the procedure how we are going to do that. So first, this is the phosphate group of this second nucleotide, and so when you will add water to the phosphate group, the bonds will break into two different OH groups by the help of dehydration reaction. So out of these two acidic OH groups, one OH group on the fifth carbon position will gonna react with the alcoholic OH group on the third carbon position of the second nucleotide. So the bond forms between these two acidic and alcoholic compounds will be 3-5- phosphoester bond, and don't be confused with these previous phosphoester bond. These two are different. That is the reason why we used 3-5- to its name, because it is 3-5- phosphodiester linkage. So let's come to the conclusion. A dinucleotide is produced with a backbone made up of alternating sugars and phosphate groups. It also has a direction, which is a 5- end with a free phosphate group and 3- end with a free hydroxyl group, as you can clearly see. Now we will see the formation of a typical polynucleotide chain. So basically, what are these polynucleotides? See, a polynucleotide chain is a long linear molecule made up of many nucleotide units linked together. These chains form the backbone of nucleic acids such as DNA and RNA, and I will explain you why. Now look at this polynucleotide chain. It's a polynucleotide chain of RNA, where this is our first sugar linked with the nitrogen base adenine by the help of phosphoester bond. Now this bond links with the another nitrogen base, which is guanine, by the help of another phosphoester bond, and similarly this chain goes up to 3-. This is started from phosphate group, which is 5- and ends up to OH group at 3-, where these sugar and phosphate acts like the backbone of this polynucleotide chain, and nitrogen base are projected more or less, perpendicular to this backbone, but faces inside this structure. I try to draw it for you just to show how nitrogen bases are arranged perpendicular to the surface of, sugar or phosphate groups, obviously. And these sugar and phosphate groups are somewhere between these lines. Okay, you just need to know what I'm saying. This is just a cartoon image. Okay. So when you reverse this polynucleotide chain from 3 dash to 5 dash upwards to 5 dash to 3 dash, it will become 2-degree double helix structure of DNA. Why it is called 2-degree? Because hydrogen bonds are present between them. Now we will see in detail all the parameters of DNA and criteria we will see. Let's move. This diagram is from your NCERT only, which trying to show the structure of DNA. These polynucleotide chains form the structural framework of these nucleic acids like DNA and RNA, which stores and transmits the genetic information in living organism. We will see in detail how they do so. So the structure of DNA was first described by James Watson and Crick in 1953. DNA had some double helix structure. Okay, so each DNA consists of 2 long polynucleotide chains twisted in a double helix way. Okay, and if you remember the structures of alpha helix and beta sheath structures of protein, they were also right-handed coiled. Similarly, DNA is also right-handed coiled. There is no difference. These strands are anti-parallel to each other. One runs 5 dash to 3 dash and other runs 3 dash to 5 dash, where sugar and phosphate forms the backbone of each strand. Now, moving towards the compositions of DNA, it has a deoxyribose sugar and a phosphate group. And thirdly, it has a nitrogen base of 4 different types of nitrogen base in it, which are mentioned on the right side. You can see with the abbreviation also: adenine, thymine, cytosine and guanine. And DNA plays important role in storage and transmit genetic information, and also acting as the primary hereditary material of the cell. Now RNA is discovered by Friedrich and it's a single strand nucleic acid. Yes, unlike DNA, they are typically made up of just one chain of nucleotide, and they are composed of a ribose sugar and a phosphate group similar to DNA. And other than that, they have some nitrogen bases like adenine, guanine, cytosine and uracil. And there is no base pairing in RNA, but at some higher level they get fold. This possible due to the complementary base pairing, and I will explain it in some another lecture. And the functions are very clear that it plays a crucial role in protein synthesis and carrying genetic instructions from DNA to the RNA. Now we have a specific term called base pairing. What is it? See, basically the nitrogen bases on opposite strands pair specifically to each other. So the nitrogen bases can't select their pair by their own. Okay, we have 4 nitrogen bases. So like example, adenine can't pair with guanine. Okay, so this way cytosine also can't pair with thymine. So these pairs are fixed. Adenine pairs with thymine only with the 2 hydrogen bond in it, and cytosine pair with guanine with 3 hydrogen bonds in it. This is called the complementary base pairing. And in RNA, base pair consists of 2 complementary RNAs, which is adenine pairs with uracil by the help of 2 hydrogen bonds, and cytosine pair with guanine as similar to DNA by the 3 hydrogen bonds in it. Always remember that these nitrogen bases should be complementary of each other. Okay, as you can't wear your shirt, like you can't button up into different hooks. Okay, if you want to look decent, then you have to button up into the right hook of your shirt. It is as similar as that. And the most common type of DNA found in nature on the basis of base pairing is bDNA, and especially under the normal psychological conditions, it is bDNA. This form of DNA with the above mentioned silent features is called bDNA majorly, and in higher classes you will be told that there are more than a dozen forms of DNA exist. Their names exist after the English alphabet with the unique structural features, and that we will discuss later on. Of course, not in this video, we can't mess it up altogether. Okay, so I am repeating it again. The 2 strands twist around each other in a right-handed helix at 36 degree, and one complete turn of this helix is about 10 base pair in it, and the hydrogen bonds between these base pair stabilize these double helix structures. Actually, it's a 3D structure, so you can't clearly count the 10 base pairs in it, but I'm trying to show you. You can see here we have, in each step we have 20 bases facing each other, and when they will form base pair we have 10 base pairs in a single one turn. Okay. And one more thing, oh my god, how many times I need to say this, that uracil is present in RNA at the place of thymine, and that thymine is present in DNA at the place of uracil. Now let's end up this lecture by this last question. So it says that we have 68 nanometer DNA. If percentage of guanine is 30, then calculate the followings. The first one is percentage and number of adenine, phosphodiester bond, phosphoester bond, and the hydrogen bond in it. This question is completely based on the concepts which you learned. So try to solve by your own. Pause the screen and then you'll match the answer with me. So the length is given 68 nanometers, and the base pair, the formula for base pair is length by distance, and we all know, and distance is 0.34 nanometer, which is given in your NCERT of class 12th, and the value 3.4 angstrom is in your 11th class NCERT. You can choose any of these two values. So we will simplify it by division and we will get 200 base pairs. And by following this we will find the number and percentage of A. So the G is 30% given in the question, and if the G is 30% then C will also be 30% by the Chargraph's rule. And by the previous question we know we have 200 base pairs in a single chain, then it must be 200 bases in each strand for the total of 400 bases. So if we have 400 bases in a single chain, then we will find 20% of 400 bases. By simple math you will get total number of A in that particular DNA, which is 80, and similarly T will also be 80, and by this you can also find the total number of G, which is 30% of 400, but it's not asked in a question, so we will not do here. What I mean to say here is you can find the number of any of the nitrogen base by following these simple steps. Then we have to find the number of phosphodiester bond in the DNA. See, if you remember, in 100 amino acids we have 99 peptide bonds in it. Similarly, in 200 base pairs we have 199 phosphodiester bond in it. So in both of these chains we have 200 base pairs, which is combinedly 400, and we'll minus 2 from the total of 400 and we'll get 398 bonds. And if we double the value of this phosphodiester bond, which is 398, by multiplying to 2, we will get the phosphoester bond, which is 796, because there is always 2 phosphoester in one phosphodiester bond. In the fourth one we have to find the number of hydrogen bond. Since we know the number of adenine from the first question, which is 80, and similarly we'll find the number of guanine also will come, it will be 120, and we also know the bonds between adenine and thymine is double bond, and the bonds between guanine and cytosine is triple hydrogen bonds. So we'll multiply number of adenine with the hydrogen bonds in it, which is 2, and we'll get 160. Similarly, we'll multiply the number of guanine with the hydrogen bonds in it, which is 3, and we'll get 360. Then lastly we'll add those values together and we'll get the number of hydrogen bonds in the DNA molecule, which is 520. And I have taken this question from somewhere, hoping for no copyrights, finger crossed. And this chapter has covered a lot of part from your molecular basis of inheritance, class 12. Just a 30-minute video lecture, which is also a good thing. Other than that, I have a good news for all, that our channel is monetized within only 10 days, which is really a great thing for us. And by just one video, it's really matters. Okay, so thank you for being till the end.